I would like to thank the organizers of the week and for giving me this opportunity to come and be here. I was sort of reluctant at first because I was supposed to be in San Francisco this week. But when Dr. James also mentioned that he's coming and he encouraged me to come over, I felt that if he can, from his busy schedule, come all the way from Cleveland, I should be able to make it from San Francisco. And I'm actually very happy that I came. This is a wonderful event. And hearing all the excellent talks and meeting all these great people has been a very humbling experience. But at the same time, the organizers put me on a tough spot because this is a very tough act to follow after such wonderful sessions by Dr. James and Lisa. I mean, I was practically tearing up. I mean, so it's I'm sure all of you were emotionally, so much emotion there. So it is a tough act for me to follow when I switch from all this patient life, no exposure to biology. When I learned about this, this was like a awakening moment for me, and that is when I decided that I'll move out of engineering to get into genomics. And then it has been a great journey for the genomics world when we were able to sequence practically anything, right? It doesn't matter what the organism is. Every organism has the same source code. So the same 4 letters make the source code of every living thing that is currently in the world and ever lived. So starting from ancient DNA, from a piece of bone, you are able to sequence the Neanderthal or the Denisovan, and you are able to practically sequence any plant, any virus. And we saw the great value and advances by which we could handle COVID and all that. But at the same time, we learned much more when we sequence everything. Hey, we are not at the top of the pyramid, right? The largest genome is a small rock fern, which has 1.4 trillion base pairs compared to 3 billion base pairs that we have. So it's 50 to 100 times more. So genomics is much more complex than we thought. And as Dr. James mentioned, we were able to at least address many of these because of the rapid development of technology. The cost of sequencing and the time needed for sequencing came down drastically, as you can see from this chart. So what took, as Dr. James mentioned, $3 billion to sequence the first genome, and then $100 million, after a few years later, fell down drastically. So now it's like maybe $200 to $300 per genome. Not in the US, they still charge $1,000, but in India you can get it for $200 to $300 for a whole genome. And I was sort of lucky to enter at the right time. You can see that bar, the Psi genome. I was there when the prices, the whole technology was going so rapidly, and that gave me a lot of chance to build a number of companies because I saw genome sequencing as a horizontal technology because it could handle many different problems or solve many different problems. So most important, of course, being human health. For us, our health is important. So human health, that is why I created MedGenome. Then I did a number of companies. I don't want to go into that. But fortunately, I could make some successful companies and create some good value. Now let us take a step back and see that. The genome sequencing gave a lot of benefits. But for health, when we go to, we could see that we could understand now many diseases which are completely a black box to us. So cystic fibrosis, we could identify the gene. We could get a treatment. Cancer, as James mentioned, BRCA1, BRCA2 was identified. But still you should think about it. The treatment took many, many years to come. PARP inhibitors came maybe 30 years after it was discovered. So there's still a long gap between identifying a gene responsible for a cancer or for a disease and getting a cure for that. But still we were on the right track. We were able to do that globally. And one significant part which I will focus on for the rest of my talk is one type of treatment through antibodies. So antibodies became, from the late 80s, antibodies became one of the significant discoveries. We all know our body makes antibodies, but antibodies that could new antibodies that the body has not seen, how do you make them and how do you use that as drugs? So this was a significant development in the US. And most of you might know, Genentech was the first company to really become a biotech. So the field of recombinant DNA and the biotech industry started off with Genentech, and a number of drugs came. So over the last 30 years, or maybe last 40 years, we have seen over 100 plus approved drugs. for various drugs, for various diseases, but mostly more than 50% being for cancer, right? And suddenly the whole world treatment modalities changed. So a number of very good examples Dr. J mentioned of how we are able to now offer precision therapy, right? So you know, okay, this cancer is caused by this gene. And so you are able to give, if the drug has been developed, you are able to give a drug that exactly hits that. So like I said, Genentech made a great progress in this. We saw a number of exciting drugs, Herceptin for any breast cancer which has a HER2 mutation. If it has a HER2 mutation that produces more HER2, then you have an immediate cure. So it's not an iffy, oh, I'll try this like a chemotherapy or some other drugs. If you're unlucky to get breast cancer, but lucky to have it because of a HER2 mutation, there is a cure for you. Similarly, Avastin, maybe not as great as Herceptin, but has been able to make a reasonable difference in many cancers. With the advent of immunotherapy and drugs like Tecentriq, things changed. We could even get much more efficacy because you are getting your own immune system to fight the cancer. Now, all this was driven by the genomics. One significant part by genomics. Of course, there are other technologies to make the antibodies and all that. Now comes a very sad part, right? Of the 100 plus approved antibodies, worldwide, even now only less than 25 are available for India. A lot. So lakhs of our patients don't have access to drugs. We saw some very interesting examples, James and Lisa's story and all that, but they had access to these drugs. Our people here don't have access to these drugs. And even the 25 odd that are available in India are 15 years later. One or two, maybe Avastin made it in 12 years over to India, because we have to wait for the patents to be done, patent life, and then we make biosimilars. So we missed out a huge opportunity, both in case of the revenue and more importantly, the treatment to patients. So India somehow missed the boat. Of these 100 plus approved antibodies, there is not a single antibody that is made in India. At the same time, India pharma was pretty good. So I am not in any way trying to say anything derogatory about the Indian pharma. Indian pharma did excellent work in vaccines, in small molecules, huge developments India made, and became sort of the pharma capital for generic drugs, for the global capital for generic drugs. But we missed out on the antibodies. So that is the first revolution of antibodies that missed out. But as Lisa was saying, or James was saying, it's not the end of the story. There is more to come. So still now, even though we have precision medicine, we are able to solve maybe only about 25-30% of the cancers. Most of the other cancers still cannot be solved. There are no right medicines. And even some of the ones that are treated and recovered, people develop resistance. So you get cancer back again. So we need better drugs. And antibodies again show the way, because we can now move forward with more exciting type of antibodies. So let us take a minute to look at the way the market has been and the various therapies. Like I mentioned, antibodies is just one mode of therapy. There are a number of other exciting technologies that are coming up. Gene therapy we heard about, Lisa spoke about CAR-T therapy. But you have to keep in mind that these are very complicated therapies, both CAR-T and gene therapy and all that. They will become mainstream, but it will take another 10 years at least for those type of technologies to become mainstream. So meanwhile, we really have to push this antibody technology, because it is already well proven. All the manufacturing facilities are there in India and rest of the world. So if you have new antibodies, it becomes very easy to produce them at scale, unlike the other new technologies that you see. The antibody drug conjugates, true, are pretty, but there you can club them into antibodies itself, because you can't make a conjugate without having an antibody in the first place. So they can be actually put into one bucket. So these two parts of this single bucket are the most important, we feel, or I feel, that going forward that we need to do. So let us take a little bit of a science class to look at how an antibody looks, especially how the more complex antibodies of the future will look like. Typically, those who have a little bit exposure of science will know the antibody is a Y-shaped molecule, a protein. But now, we have the technologies to add more arms to it, right? So you can make it bispecific, trispecific, and multivalent. So multivalent meaning multi-specific, multivalent antibodies. That means these antibodies can hit multiple targets multiple points of the same target, it can hit multiple targets as well. So why is it so important? Because cancer is a deadly disease. And cancer is not like, not easily curable. And typically, except for that 25% of cancers, it is not driven by one mutation. There will be a number of genetic mutations that play a role in the cancer. So if you are able to add more targets to your antibody, you are able to hit multiple places at the same time. So this improves, so you have a choice now, or opportunity now to improve efficacy, because you can hit more targets. You also have a chance now to reduce toxicity. How? Because one of the biggest problems of many drugs, many antibodies as well as other drugs, is the toxicity, because the drug goes to other places other than cancer and kills your good cells also. But now, let us say you have a target which can use to kill cancer, but you have another target which the cancer produces. But if killing that won't cure the cancer. But fortunately, that antigen is expressed more in the cancer cells than in other cells. So now if you have multiple arms in your antibody, the second target will act as a beacon to take your drug to the cancerous cells only. And only very little will you be hitting the other cells. So these two are very, very, very effective mechanisms by which you can actually go back and save or bring back many of the drugs that failed clinical trials before. There are hundreds of drugs that have failed clinical trials. And mostly it will be because of these two reasons. One, the efficacy is not that great to cross the bar. Two, the drug was great, but it also kills the patient. But at the same time, you have to think about it. Those drugs were actually really good. Otherwise, pharmaceutical companies would not have spent millions of dollars to take them up to phase 2, phase 3. So these are the low-lying apples that we can take and try to solve them, resurrect them using engineered antibodies. So engineered antibodies are one of the wave of the future as far as this technology goes. So this is a typical example that I want to do. VEGF is a very well-known drug, anti-VEGF. I talked about Avastin, which is an anti-VEGF, and we also talked a number of times in the previous talks about PD-1. So now these are both successful drugs separately. Now if you combine them together, they become much more efficacious. Right? And very little effort in clinical trials all are needed, because both are approved. So you just have to make sure that your combination does not create any other problem, the single one. And the cost comes down. So already in the clinic, both are being administered together. I mean, two different molecules put in together. But that increases the cost, also increases the load on the patient, having two drugs coming. So a single molecule therapy with these two arms, 4 arms basically, but hitting 2 targets was one of the first ones to be produced as a bispecific. So Dr. JM also talked about the bispecifics coming along. So this is one of the first bispecifics. And now there are about 6 or 7 bispecifics approved. There are about 100, 150 odd in the clinical trials. But look at the phenomena in the second part of the slide. China has overtaken even the US. So the number of bispecific and trispecific antibodies being developed is much more in China than even in the US. And many of I know people might think like, yeah, China, we always have heard bad stories about the Chinese quality and all that, which is true even now to some extent. We have seen in the US many of the results that are shown in Chinese publications. Same drug, when it is brought to US and repeated, many times it doesn't work out. It doesn't show that. But that is past. Now you can see that Summit made a big announcement by licensing one of the molecules from China, VEGF PDL1, for billions of dollars. There has been at least 4 or 5 deals in the last 6 months of various American companies going to China and licensing that. And we are nowhere in the world, nowhere in the map. So if you want to achieve the Atmanirbharta, which I know I screwed it up, but but you get the point. You must have heard the Prime Minister saying that a number of times. So we can't afford to miss this opportunity again. So what can we do? What can all of us do? There are clinicians, researchers, biotech and pharma, and the government. So clinicians are at the front line, right? They're the most important cog in the whole chain. So India, we need to do genetic testing much more. We need to integrate the data that you get from a genetic test into the electronic health record. Right? And because often that is not done. Genetic test is done, the genetic report may be stored, the test report may be stored, but the critical piece of information of the mutation, gene and the mutation, has to be stored properly and linked to the patient's electronic health record. And typically the samples are thrown away. Maybe the diagnostics company might hold it for 6 months or something, but other than that, nobody saves the biospecimens. So those need to be stored. Right? And all the clinical data is so important. So the data needs to be collected uniformly across different hospitals. Typically now in India, every hospital is in their own journey. There is very little uniformity. Forget even data collection. Even in treatment, there is very little uniformity. So that protocols are missing in India, so those need to be put up. The clinicians themselves have to take the leadership. Most of the clinicians, as oncologists I have seen, and other diseases also, are very passionate about their work, but I think they need to I don't know, we don't have the right committees here, I think, which can enforce uniform protocols, but that will need to be done. The researchers will need to do a more thorough, more extensive sequencing of these samples once you have these samples in place, because the clinician might have looked only at 1 or 2 genes of interest where a medicine can be applied. We, for developing new drugs, need to know the mutation profile. And this varies tremendously across populations. So even in the future, when new drugs come, if it has to really satisfy the needs of Indians, we need to have our data. If we don't have our data, we won't know which drugs to develop, which should be given priority for our population. So that has to be done. And a lot more technologies are now available to develop very good cell lines from human patient-derived cell lines. We can make organoids. So all these can reduce the cost and agony of doing too much studies on animals, and all those can be reduced. And we can speeden up the process of bringing these drugs to the trials and to the thing. So pharmaceutical companies, like I said, Indian pharmaceutical companies have great manufacturing facilities. They did a lot of progress in biosimilars. So all the manufacturing facilities for antibodies are already there in India. We just need to use the new technology and make the new molecules. And also, we need to strengthen up some of what the researchers I mentioned overlap with the pharmaceutical companies too, because they have their own researchers. So we need to build the tools to speed up the process from a creating of an antibody to getting it to clinical trials. Now the government is the big gorilla in the room, but unfortunately, all these first 3 things we can do, or fortunately we can do without much involvement from the government. But finally, at the end of the day, for the clinical trials, you need the regulatory agency's approval. So very surprisingly, till about 4-5 years back, there has not been a single phase 1 trial approved for an antibody in India. So our organizations did not even have the necessary forms or the protocols. Only in the last 3-4 years, we have started with a very few. Still, it is very few, and it takes a long time. So the government really needs to step up. They really need to make that much more agile, get it faster, and also rethink the whole concept of clinical trials. In the sense that, especially when let us say you are combining 2 known molecules, why should have an extensive clinical trial with thousands and thousands of patients? You just need maybe 10, 15, 20 patients. And as we go along with more precise therapy, when you are having more targets, we should not have too much extensive trials. So those are the things which not what I am saying, I alone am saying. There are a lot of people across the world saying for that. So we can get ahead of the curve, India, if we get our regulatory agencies to look at it more proactively and become even more better than the FDA in the US. And we have a lot of scientists of Indian origin in different parts of the world. So I was very lucky to work with Dr. Sachdev Sidhu, a person originally from Punjab, but born and brought up in Canada. And he has been one of the world's expert in antibodies, engineered antibodies. So he has moved out of University of Toronto. Earlier his career started at Genentech, and then he was in University of Toronto for 15 years, and now he has his own company, and he has the best set of antibodies in the world. And also his antibodies are such a way that they are very modular. So it really helps for those new type of antibodies that we want when we want to put them together as a Lego blocks and create new antibodies. So Dev himself has become an entrepreneur because we are so unhappy at the bigger companies, larger pharmaceutical companies, not really pushing many of the new molecules forward at the pace that he wanted. So he himself created 4 companies and has been very successful. One of his companies, Antlara, it's for regenerative medicine. You know, the deer with the horns that grow antlers. So they call it Antlara because they have a set of molecules that can regenerate, antibodies that can regenerate various tissues. So one of that was licensed by a company called iBio that regenerates, the retina of the eye. So a completely new pathway. And within 2.5 years, they were able to bring it to clinical trials. And within 6 months, they got acquired by Merck for $3 billion. So just to show that how powerful these type of new molecules are, new antibodies are. And the same company, Antlara, had a few more molecules on the same, regenerative approach. And Roche last month bought the whole thing. Roche is a parent company of Genentech, as you all know, the Swiss company. So they acquired all those antibodies. So we now, okay, I'm not a preaching guy, so I am more of an entrepreneur. I've been an entrepreneur all my life. So my goal is to see, hey, what can I do? So what I have done is that I have tied up with Dr. Sidhu, and I have brought many of these new antibodies down to India. So we are now working on to create a lot of combinations of these antibodies to create very powerful, multi-specific, multivalent antibodies in a mass scale. So I'm not like not a drug company trying to take 1 or 2 and take it down up to the clinic. That will take too long and needs too much money. So what I and my team are trying to do is that we're going to make a lot of antibodies, but do only to the early part. So get the initial in vitro work done, the initial lab, some animal models, and bring it to a pre-IND level where then we have larger companies both in India and global take it over, and they can, then this can be spread across a number of companies, and they take it and bring it to the clinic. So I hope that in the next decade, India can, put a flag in the map and say that, we also have first-in-class, best-in-class drugs. So that is the hope. I'll leave a little plug in the end. This is a book a couple of years old, but that talks about the challenges Indian entrepreneurs face. But I also really highlight genomics and biotech industry and sort of predict the bio wave. The next bio wave, I think the next 10 years, 20 years, we'll see a huge bio wave in India. And we'll see the even up to the synthetic biology applications that Jay briefly mentioned. So it's just not therapeutics. In biomanufacturing and synthetic biology also, we'll see great developments. Thank you.